The floor is yours.
Thank you very much, and thank you for inviting me. And it's been really nice to be here, and I'm excited to share some of the work. And Professor Kamada's group has done some really excellent experiments in remote sensing that I've learned a lot from, and the data that you guys have created and published has been really a valuable resource for our group and our understanding. And I really appreciate both the invitation and also all the work that you have done over the last many years. It has been very instructive. So I'll give a little bit of introduction about myself and my lab, and then talk a little bit about the California context and why we do what we do, and then we'll go into the details of the presentation for today, which is about using remote sensing to understand the nitrogen nutrition index, which some of you may be familiar with. I'll explain a little bit of those concepts in case you're not. So it looks like the text got a little bit shifted here. That's okay. The main overview is related to nitrogen use efficiency. And how does nitrogen use efficiency relate to nitrogen nutrition index? And then how can we use remote sensing tools that already exist to form a nitrogen nutrition index? And I know that we've done some work here and elsewhere on that. We have a really broad program in California. As Professor Kamada said, we coordinate all the applied research related to winter cereals in the state of California. So that involves a lot of field work and field research, field-based research. We do late-stage variety testing at the end of the breeding pipeline to understand genotype by environment interactions. And we apply precision agricultural tools both for phenotyping in the context of genetic selection, as well as in the context of creating understanding and tools for precision management in the field. We present a lot of that through interactive web tools. So in addition to being agronomists, we increasingly are becoming software developers and data analysts, and that's, for a long time, agronomists in the agricultural research context have been, in English we say, a jack of all trades, a master of none. And as the tools increase, the number of trades that we have to be proficient at increase. And I think our job as agronomists is really to be able to have a conversation with a breeder, have a conversation with a modeler, have a conversation with a farmer, have a conversation with a data scientist, and be able to integrate those parts into a whole picture and come up with good questions, understand what the needs are from the different perspectives. So we work on that pretty broadly. California is a really diverse agricultural production region. If you're not familiar with it already, a typical farm will grow somewhere between 12 and 20 crops. Across the state, we have over 120 different crops grown. The crops grown are highly diverse, from really high value production, fully irrigated in the summer season, to really low value production, rain fed in the winter season. I primarily work with winter cereals, so wheat, barley, triticale, a little bit of oats and forage mixes. Those crops are grown in the wintertime. They are widespread. This map is indicating all the places where, let me see if I can find the pointer here.
The center button.
Yeah. All the places in California where small grains are grown, and that's most of the agricultural production valleys in the state. I always joke, if you ask a farmer in California if they're a wheat farmer, you say, "Are you a wheat farmer?" They'll all say no. If you ask them, "Do you grow wheat?" they all say yes. So it's a small part of their gross, and so we get a small portion of their attention, because the things that are limiting their system primarily are water, labor, land values, and when you're competing with tomatoes or with almonds or with vineyards for the same resource base, you have to be highly efficient to make the grain system productive and to make it efficiently profitable. So we have really highly variable conditions in California, and that results in really highly variable management approaches across the state. I'm going to zoom in briefly. This is the San Joaquin Valley, which is the main production region in California. It's an irrigated portion of the state, and it has high precipitation variability. So on a 10-year average, this is in millimeters, and this is the driest year across a 10-year average, and this is the wettest year across a 10-year average. And so we have a lot of extreme variability, both spatially and temporally. And then those spatial patterns change across the years. So if you're in the summer cropping system and you have very little to no rainfall and it's all irrigated, this doesn't mean quite as much, except how much is in the bank, either at the reservoirs or in the groundwater. But if you're managing winter crops, which we do, this is a key component, because we have unpredictable and highly variable water moving through our soil system. So that has big impacts on productivity and nitrogen use, which we'll talk about. So as a result of that variability, the approaches to water span the entire range, from rain-fed to fully irrigated to everything in between. Predominantly, we have fall-planted wheat, barley, triticale with a weak fertilization requirement. It's winter wheat in practice, but genetically it's spring wheat. And we do actually have a little bit of true winter wheat up in the north that has a fertilization requirement where it snows up here. But we also have some spring-planted wheat up in this area, and the small grain production, the wheat and triticale and the barley, is grown both for grain and for forage. There's 40 million people in California. There's about 1 million dairy cows, and they eat a lot of forage. And the alfalfa in the state and the maize in the state has primarily been the ration for the dairy cows, but with water regulations in the state, squeezing the availability of irrigation in the summertime, more of the feed ration is starting to come from the winter cereals. And so I've been working at UC Davis in this capacity for the last 10 years. In my 10 years, we went from about 60 to 70% grain production and 20 to 30% forage production to the opposite. We're now at about 60 to 70% forage production and 20 to 30% grain production. So the outcomes for the harvested product are very different, and it's cut off there on the bottom, but the overall yields range from some of the lowest grain-fed yields in the world, about 1,500 kilograms per hectare, to some of the highest in the world, 10, 11 tons per hectare. So we have a huge range of productivity possibility, and that's an important thing to understand when you're looking at our data. We have a really diverse data set. So within that context, we measure late-stage varieties and elite lines and measure them in a lot of contexts. We piggyback those trials with agronomic trials that are familiar to probably you guys with irrigation gradients or nitrogen gradients. And we do that all over the state. So we have a really extensive trial network. Most of these are on farms, but we have several stations throughout the state where research and extension centers where we can do more controlled experiments. There are 4 main ones that we work at. At those controlled sites, we have low water, low nitrogen, and then maximum productivity zones in a subset of our treatments. And then within the trial, we have a lot of different genotypes. So those little squares are replications of genotypes under different nitrogen and water management conditions. And then we've been layering for many years on top of that multispectral UAV-based reflectance measurements using primarily MicaSense cameras that measure 5-band blue, green, red, near infrared, and red edge bands. So we have probably about 40 to 50,000 observations pairing a multispectral measurement or multiple multispectral measurements in season to end of season productivity and protein content from grains. We have less in season, and we'll talk about that in a second when we talk about our model development. But we have a pretty extensive trial network. So we try to take that information and turn it both into scientific information as well as practical information for making management decisions on a farm. And that could be from a farmer themselves, or there's an extensive advisory service in the private industry in California. So a lot of our customers are people whose job is to give advice to multiple growers of wheat across the region. And so those customers, I think maybe it's because I'm not in the full view here.
Go with it. Go to the other
Oh,
screen.
okay.
There you go.
I can just go to the website, see if it doesn't work. Those customers receive our information through a website that we curate, update, and oh yeah, there we go. And so they come here for information about educational events, or making decisions about nutrient management, making decisions about what variety they should be choosing. And one of the things we've really worked on is upgrading that kind of information from static to dynamic formats. So incorporating environmental data as it's happening in real time at the location where the farmer's making the decision. So we have a number of these. The one that is the most expansive and incorporates the most environmental data and most complex agronomic models is our nitrogen management module. In this module, it is there we go. It allows the farmer to select their location, the date range of interest, and based on some agronomic models that are running in the background, we're able to give them a site-specific estimate of what their nitrogen use looks like at that moment, how that compares to historical averages. We allow them to fix our models. If our models are not correct, they can slide this around and say, no, we're not at that stage of growth, we're later or we're earlier. And then based on the stage of growth and some site-specific measurements that they take, we'll maybe talk about, we can then run a model that gives them a very specific recommendation of likely yield response or protein response based on the measurements they took. And the measurements that we have them take are reflectance measurements from either leaf or canopy or drone or satellites. And then we express that as a ratio. And then the soil nitrate quick test. We worked a lot on integrating a simple test with site-specific soil information so that they can translate that into a fertilizer equivalent in an easy way. So those are the kinds of things that we try to take all of our information and put it into a package that helps the farmer make a specific important decision. And we have some other ones that I won't show you right now, but we could explore a little bit more. But importantly, this really helps us to, here, I think,
No, yeah. Go down.
Okay. That really informs us about what we should be researching. So this is represented as a circuit, and none of these nodes are closed. A farmer often will make an observation that leads us to better understand or question some scientific truth that we thought we knew. Likewise, they're identifying problems that we haven't been able to solve specifically enough to be useful to them, and that helps inform our research. And of course, we're interacting with other research groups like the work that you all do and other parts of the world. And so all those nodes are circular and helping us to always sort of revise what we're doing and how we're doing it. So within the broad context that I just gave you, nitrogen use efficiency is really a key target for what a farmer does on the farm, what we do in a research context, and how our farming systems end up impacting the rest of the world outside of the farming community and the farming environments. I think you guys probably already know some of these types of stats, but I'm going to step behind on this and that.
Go to the other screen.
Oh, I have to click it there.
There.
Yeah, sorry about that. But nitrogen use efficiency in major cereal systems averaged across a lot of different landscapes is not that good, about 40%. And that means that a lot of the fertilizer that we apply, we've had about a 3-fold increase in fertilizer over the last 50 years, about a 2-fold increase in food, which is a good outcome, but our marginal returns to increases are lower and lower. Within the California context, the models that we use, the models that we develop and try to show farmers how to use, they achieve much higher nitrogen use efficiency than that. And I've seen similar models in the Midwest, similar models here in Europe. When you get into the context of a specific region, a specific cropping system with agronomists who know what the ground floor looks like, you can achieve much higher nitrogen use efficiency if you take enough measurements, if you take the right measurements at the right time. But writ large, we're not doing that. And we are aware of the consequences in terms of water pollution, in terms of air pollution, and Europe, as California, has strong regulatory apparatus in place to try to mitigate those negative environmental consequences. So farmers in California they're monitored for how much nitrogen they use in aggregate at the watershed level, with the threat of if compliance is not achieved, then they could be monitored at an individual level. So we are regulating with primarily a punishment approach because it's cheaper than an incentive approach. What I want to get to, and we'll talk to about at the end, is how can we restructure how we measure nitrogen use efficiency so that we might be able to have some incentive approaches to achieve nitrogen use efficiency. So NUE is a really unifying concept at the farm in what we do and at the environmental level. But we're just not picking these low-hanging fruit at scale and across the globe and across regions. So why is that? Well, I think that part of the problem is that we don't have a common target. And if you are in a crop management setting, what does nitrogen use efficiency look like to you? Probably means how much yield you get for how much nitrogen you use. If you're in a physiological research lab, what does nitrogen use efficiency mean to you? It probably means something different. How efficiently is the nitrogen in the plant being transferred into the outcome that you're trying to measure? If you're in a crop monitoring situation, nitrogen use efficiency to you probably means something at a really large scale, at a watershed scale, or at a state scale, or at a national scale. And so those budgets are based on very different spatial and temporal scales. And so they mean different things to different people. Some recent work that I was involved in, I do a lot of work with meta-analysis. Professor Kamada mentioned the no-till work that we've done. And one of the ways that we come up with questions to work on in an actual field is by first looking at what the broader scientific literature says about something. Recently, we compiled a lot of information about nitrogen response curves, primarily in wheat and maize. There were some other minor cereals in there too. And we calculated two metrics, the agronomic optimum and economic nitrogen rate. So you'd be familiar with this in a typical study where you have different rates of nitrogen fertilizer and you measure grain yield. You can locate where it stops making sense to apply more nitrogen, and then that becomes your optimization point if you're talking about from a plant physiology perspective or from an economic perspective based on the price of the fertilizer. And then you're probably familiar with approaches like nitrogen budgeting. This is the EU framework, and we have similar frameworks in California where inputs and outputs have to be more or less in balance with a little bit of fudge factor for cycling that goes on in the soil. But big picture is when you actually take those kinds of aggregated metrics and apply them to an individual field, they rarely are correct. In fact, only 35% of the time are they correct. Most of the time they were wrong because they're based on aggregated data, and individual fields happen in a disaggregated way. So they miss things like soil nitrogen supply of that individual year with that individual interaction with rainfall following this particular crop rotation and all the interactions therein. You have to have some soil nitrogen incorporated into your understanding to be able to have a robust nitrogen sufficiency method. So we're not really aiming at the same target when we're talking about nitrogen balance, or if we're talking about fertilizer recovery efficiency or nitrogen utilization efficiency. These are all adjacent concepts, and they're all somewhat overlapping, but they're not the same. So I think that nitrogen nutrition index has potential to really take us to a more unified concept. And there's been a lot of work here in Europe, primarily here in Europe, on the nitrogen nutrition index. And I think we're at a stage where we have the measurement platforms, and I think we might have the know-how to be able to predict the components of the nitrogen nutrition index, which from a modeling perspective are actually not too complicated: biomass and nitrogen concentration. And if we can do that from existing sensing platforms like Sentinel-2, then we could have a unified measurement and be comparing apples to apples in all the different regions of the globe. How you hit that target is probably going to be different in California than in Spain, than in France, than in the Midwest of the United States. But if we all have the same target, then we all can measure success on equal footing. So I think we need to work on that. I'm just going to ask a real quick question. Are you guys all familiar with the nitrogen nutrition index? Raise your hand if you're not familiar with it. I've never heard that before. Okay, so
you can go. I mean, some of us we are familiar with. We are familiar with the nitrogen index.
okay, I won't spend too much time on that. If everybody needs that one. Okay, so if you're familiar with this, you know how the curve is made. Different rates of nitrogen measured at different times. Importantly, though, these two measurements measured in season aren't that common. We do that big meta-analysis, and you go look back in those papers. So many of us, myself included, we measure grain nitrogen, we measure grain yield. But how often are we measuring something in season? Very few studies are measuring things in season. So it's very possible to create this kind of curve, but it's not very common. And some work by, so you all know that, so we have the critical nitrogen dilution curve here. And one of the things that caught my eye in the recent past, reading your all's paper and then reading a little bit more about the nitrogen nutrition index concept, David Mikowski and Ignacio Ciampiti, who used to be at Kansas State, is now at Purdue, they were working on the critical nitrogen dilution curve through meta-analysis. And so they were taking all these studies where use and everybody was reporting slightly different versions of the curve, and they were trying to use a Bayesian approach to estimate where the likely curve was after you consider all the empirical incorrectness of each individual study. And so what they came up with was that that curve is actually more stable than previously understood. I took their curve, I'll show you this in a second, I put it into some of our data, our long-term nitrogen deficiency sufficiency data, and it fit perfectly in just in our measured data. And so that got me really interested and motivated to start seeing the extent to which we could predict these two components from measurements that we'd already taken. Let's get cut off too. So I'm glad you guys know the concept already, because it's actually getting cut off. So this is exciting because I think we have the ability to create this unified target. And while we've been doing the work that I'm going to show you primarily here, we've also been deploying meta-analysis and asking the question, to what extent can we use the same metric to quantify ecosystem services and associated trade-offs? So we did that in two forms. We tapped into some of that bigger meta-analysis data that we have, and it's like this. Let's see if I can move it so you can see. There we go. We asked, to what extent does NNI serve as a good predictor of nitrous oxide emissions? And from a plant physiological perspective, it should be a good predictor, right? If your plant is in luxury consumption, what does that mean for the soil environment it's in? It means that the soil environment likely has the conditions where a little bit of moisture differentiation is going to lead to gas off. And so if you are sufficient or below the NNI of 1, you're likely not going to have very much opportunity for nitrous oxide emissions. So we tested that in this data set, and we came up with a probability that emissions exceeded what are considered background emissions for cereal crops in cultivated production system, which is a little bit more than 1 kilogram per hectare N2O. And so we calculated that probability, put it into the context of an NNI curve, and you can see, not perfect, but the high probability emissions tend to be the NNIs that are really high, both in the context of the curve, as well as in, this is just using the probability as the y-axis and NNI on the x-axis. These are the absolute measurements of N2O emissions. If anybody has worked with N2O data, it's like finding a needle in a haystack. It's always highly, highly variable because of the way it's measured. But you're trying to get a signal from something that happens in a really short period of time that's 3 orders of magnitude less than the whole nitrogen in the system. So this is very typical N2O data. But importantly, all the really high values and high probability values are happening after NNI 1, which is what you would expect from a physiological perspective. So that's a hopeful development. But then from a farmer perspective, and I'm sure if you guys have worked with farmers, you see the same thing. A farmer can apply just the right amount to achieve good nitrogen use efficiency, and probably in 4 out of 5 years have no yield penalty. But the reason that they tend to apply more than they need is because every 4th or 5th year they actually do need the extra nitrogen. They're even in highly productive and precise systems that I work in, their yields vary by 15 to 20% year over year. And so when you have that really high yielding year, if you do the math on the cost of the nitrogen at 15 to 20% excess, losing 15 to 20% of yield 1 out of every 4 or 5 years is more costly than paying for that extra nitrogen every single year. So it makes economic sense for them to be applying more nitrogen than they need. They're worried about yield loss. And so can we use NNI to quantify that same thing? If you have NNI measuring N2O emissions risk, alongside measuring the risk of yield loss, then that same metric could tell a farmer, yeah, you did actually lose yield here. You lost the possibility of yield, or you did not. That can be turned into markets. We'll talk about that at the end. But you need the same measurement to be telling you about the environmental risk that's telling you about the risk to the farmer. It has to be the same measurement. And it looks like NNI is doing a fairly good job of that as well. Across a lot of environments, NNI was predicting yield loss as effectively as essentially going and doing an N-rate trial at all of those locations. So once you incorporated the multiple nitrogen rates that were in these trials by the site-specific year effect, NNI was getting about the same amount of information. So that was encouraging, also really robust across a really wide range of soil nitrogen backgrounds. The blue is a highly responsive site, low background mineral nitrogen. The red is not as responsive to the site. Importantly, though, on both of these, we were calculating NNI from a post hoc measurement. So we were having to translate from grain yield, grain nitrogen, and move backwards to what NNI would have been at anthesis. So we're making some assumptions there that probably are not correct if we saw a lot of differentiation in nitrogen utilization, efficiency, nitrogen allocation patterns during grain filling. That brings us to, why don't we have more NNI data? Well, it's really hard to measure. And so we had to use the data we had, which was a bunch of grain data. And the phenological timing of that measurement really matters a lot. You've probably seen that in some of the NNI literature. Anthesis seems to be the ideal time to measure it. You're at the end of the nitrogen uptake curve. You're moving into distribution and nitrogen allocation. And the interactions with drought and other abiotic or biotic stresses will certainly play out there. But at that moment, you at least have the full or almost full nitrogen uptake in the total above ground biomass. So I kind of already said these all aloud, but can we predict these from remote sensing platforms? How well do they translate then? If so, how well do they translate to a satellite-based measurement? Okay, so then we'll move into the heart of this. Before I talk about the specific methods, I just want to give you the key points of the methods, because we're doing something that's a little bit unconventional here. This is what happens when you're not actually a remote sensing person and you start messing around with remote sensing data, trying to get an outcome. Sometimes you think of something that somebody already tried. Sometimes you're like, well, why didn't they try this? So two things about this. One is that I know that the phenological measurements matter. We see that a lot in the nitrogen work that we do. Knowing the stage of growth is the biomass and the nitrogen content is subordinate to the stage of growth. From a plant signaling perspective, we know that those are hierarchically nested. So phenology is going to matter. So we incorporated phenological information into our prediction framework. We've worked really hard over the years across different cameras, across different experiments, across different NIR machines where we're measuring our nitrogen content in our tissue to calibrate our data. Because if we're going to put it all in just one big group, we have to know what it means in absolute terms. So we always, I'm kind of obsessive about calibrating our data. So we have really well calibrated nitrogen concentration data. I think if you were trying to get an absolute measurement, like what we're doing here, from poorly calibrated data, it wouldn't work very well. We have a lot of diversity in our prediction features. I'll go into that a little bit more. And this is a little bit unorthodox. There is value in knowing what a specific wavelength and reflectance value or indice means in terms of water stress or in terms of nitrogen stress. That research is essential. It's widespread, but it's not always needed if you're not trying to make a direct relationship between the indice and the outcome. So we really widened the prediction features pretty dramatically. And we'll talk a little bit about that. And then, as I mentioned before, we really worked to follow a plant physiological framework when we were making the nested predictions. So these are the key points. I'll explain them a little bit more as we go through. This is giving you an idea of the scale of the data. You already kind of know about California. I spoke about it already. We have Mediterranean climate type, highly diverse. We have a lot of soil types, and we measure lots of genotypes across a lot of different variations in these factors. So we have a pretty broad number of biomass observations across a wide range of productivity in a lot of different fields. Same goes for the nitrogen content, including places where we know there was managed stress, and we've taken a lot of phenological observations on top of these same observations. So we kind of backed into this data set. So if you're designing this to get this outcome, you probably want to design it this way, but it is what it is. So that's how we kind of came to this data set through pursuing other means, but it wanted to know whether it could serve us for this outcome. So these are actually measured values, okay, before we get too far. This is nitrogen content that we've measured. These are field day combinations. This is biomass that we've actually measured. This is the universal critical nitrogen dilution curve reported by Ciampitti and Mikowski. And so really across a wide range of conditions, that curve seems to be well suited to the measurements we've been making, okay? We have a lot of different genotypes in there. We also have diverse crops in our system, so we just threw them in there. The C3s, the carbon to nitrogen ratio at a cellular level is pretty similar. So if we're actually talking about carbon to nitrogen ratio as a stable predictor, it shouldn't be all that different across those crops. If you're going to move into rice, maybe. If you're going to move into corn, definitely, because it's C4. But we thought, you know what? We want to represent as much diversity as possible, knowing that the carbon to nitrogen ratio at the cellular level between these is pretty similar. So that resulted in about 1,900 observations, multiple UAV flights, multiple site years. Again, blue, green, red, red edge, near infrared. So this is the key thing from this one, two things. This is the scale of our typical extraction. So that's about 10 centimeters. Some of our earlier data was at 15 to 20 centimeters, so it's not perfectly consistent. It's between 10 and 20 centimeters are our pixel sizes from our UAV flights. We could have gotten smaller, but we wanted to better represent the canopy, the population of plants, than trying to get in features that were sub-plant level. So our pixel sizes are between 10 and 20 centimeters on the side. And then we just collected all of the indices that you could calculate using those 5 bands. So that includes the NDRE, NDVI, MCARI, but a lot of other things. And then the other thing that we did based on our observation of lots and lots of fields with nitrogen deficiency or water stress, if you guys have been in those fields, which I know a lot of you have, what you see is spatial variability. That's always an indicator of abiotic stress. And so we said, why aren't we using spatial predictors in this? We have the ability within a grid to make a coefficient of variance. We have the ability to calculate a standard deviation of an indice. We have the ability to calculate a spatial variance of red relative to blue. So why aren't we doing that? So we derived a bunch of spatial prediction calculations that were representative of the block of plants and included those as prediction features. This is pretty standard, using XGBoost, tenfold cross-validation. We first took a 10% holdout, and we were using the tenfold to develop our features and figure out where we stopped improving on the root mean squared error. And then that feature set was fit on the entire 90% and then predict that 10% holdout. We did some leave one out, but we had a couple of sites that were really imbalanced. And we needed a couple of the sites where we had taken a lot of biomass and nitrogen concentration. So we couldn't really do it through leave one out. But the leave one out that we did, accepting those 2 sites, had pretty similar outcomes as this 10% representative holdout. And like I said before, we just ignored multi-field linearity. You know, these machine learning methods are really good at nonlinear relationships, They're really good at handling weak predictors. If we're not trying to understand specifically what's going on between NDRE or MPARI or any of the other indices, we're just trying to get the best biomass prediction, nitrogen concentration prediction. We're just going to ignore multiple linearity. So we just ignore it. Like I said before, we know that biomass and growth stage are highly related to each other. And so we created a prediction scheme where we first predicted growth stage. We fed those growth stage predictions into the predictor set. Then we predicted biomass, fed those predictions into the predictor set. Then we predicted nitrogen content. And that's the way that the plant signaling occurs. Okay. So then this is our holdout data. This is our observed biomass, predicted biomass, observed nitrogen content, predicted nitrogen content. And that root mean squared error and relative root mean squared error that we're getting is pretty close to our error in our measurements themselves. So if we go out and measure biomass across 4 replications in our fields, we get about 20% average error. So it's a little bit worse than our average measured error. This is about the error that we get off of an NIR machine. So we're getting pretty good predictions on the holdout data, but a lot of features. And importantly, both of these had a lot of features, but about 40% of the features are these spatial features. This is not biomass data, this is grain data. We have a lot of this data where we measure grain and grain nitrogen content at the end of the season. And we had flights at anthesis over these same fields. So this is our predicted nitrogen content, our predicted biomass content for a year where we had either applied fertilizer or not applied fertilizer. Okay, so this is at anthesis, this is at flowering. This is another year. The red outlines are where no fertilizer was applied, the black outlines are where there was. These are different genotypes in those trials. This is the next year. And so what you're seeing across these years is that the background mineralization is different, the response to nitrogen is different, things that you understand intuitively. But the framework here of the nitrogen nutrition index is contextualizing the biomass to nitrogen ratio well, the carbon to nitrogen ratio well, and creating a normalized measurement across those. So that's all of them combined. And you can see that there's a few of the unfertilized nitrogen up here. Maybe there's more background nitrogen in those soils, or those particular genotypes have low, have different nitrogen use efficiency patterns. But you don't really see any of the fertilized squares down below that critical nitrogen dilution curve line. And so if we take that grain yield that was measured and multiply it by the protein, so we get grain nitrogen yield, and compare it to our above ground nitrogen anthesis that we predicted from those remote sensing measurements, we get a very, very strong linear relationship. Not perfect, but a lot of variation here. But overall, root mean squared error, similar to what we were getting in our holdout data, but knowing that we had to correct the predictions in anthesis by 2 things. One, nitrogen harvest index, not all of that accumulated at anthesis was going to go to the grain. So there's a known relationship there. But of course, how that plays out across the different environments probably matters, how much nitrogen stress there was, how much water stress and the interaction therein. And genotype obviously have a say in that. The other is possibly post-anthesis ammonia loss from leaves. We were wondering where some of this nitrogen went, and I actually, to be perfectly honest with you, was not very well aware of how large of a scale the ammonia losses can be from leaves. But there have been old studies, but several studies using isotopes and trying to really track that, and actually finding that, especially in highly fertilized conditions, you can get up to 30-35% lost post-anthesis oxalates. So it varies across the nitrogen gradient. So this is something that I learned trying to figure out where that nitrogen was going. But once we took that model and applied it to our data, it basically corrected the difference between where we knew it was going to be lost to the nitrogen harvest index and where the other nitrogen was going. I think might be ammonia loss, which is very hard to measure. And we don't have a lot of good data at anthesis and at grain filling. So that one factor that was kind of new to me as we were starting to do this. So that was working fairly well using the UAVs, and I thought, okay, how badly is this going to break when we try to get it to the satellites? Because we really want ultimately to be able to do this at a satellite scale. And so we compiled 2 data sets, one from California over a long-term experiment where we have about 0.4 hectare replications, and there's water and nitrogen gradients. Not perfect for this, but pretty good because we get some real extreme low nitrogen and some high nitrogen outcomes from this trial. It's not perfectly managed, the productivity outcomes aren't great, but there's a cover crop component to this, and all the yellow squares or partially yellow squares have wheat in them. Those are maize. So there's a good long-term data set and big enough for us to extract sentinel pixels from and test it. And then the other was your old data set. The really detailed measurements taken here across 2 water gradients, 4 nitrogen levels, 2 seasons, and the multiple scale and both spatially and temporally, really unique data set, really valuable data set. And the fact that you guys are willing to put that out into the world and share it and let other people learn from it is really admirable. I mean, there a lot of people talk that game, not a lot of people walk that game. And so I really can't say enough how much I've learned and appreciated you guys' ability, not just to publish the papers, but also to put the data out there so we could learn from our data as well. So the one thing that I want to note here that's not showing up, I'll try to pull it up. I have a great model for growth stage in California and weather sources, and we've calibrated that extensively. I don't have a good model for that here. I know that the weather source can really matter because if you're going to accumulate temperature over a long period of time, your thresholds become very sensitive to the source of weather data. So I didn't really try to predict the growth stage here using my California models, but you guys did report the growth stage in your papers. So then I was able to take those growth stage and just set the model at 65, say about 65 for anthesis, and then run the rest of the model from there. So the one piece that's different between these two is that I just kind of entered 65 manually for your all's trial. Okay, so then these are the predicted nitrogen and predicted biomass contents. You'll notice that I multiplied each of them by 0.9, so we had about a 10% reduction. I'm going to talk about that next slide a little bit more, but we did see an overestimation across the board. But in this case, it's about 20% different from what we were getting from the UAV. So I just split the difference, half in the nitrogen content, half in the biomass, and that's something that I'm curious to talk to you guys about and explore more. But basically, all of the red in this across the 2 different trials are where fertilizer was not applied. All the blue is where fertilizer was applied. The outlines are indicating no or yes irrigation. And so again, that's our critical nitrogen dilution curve. These are predicted values corrected by about 10%. And so across a lot of different site years, at least from a nitrogen perspective, that curve seems to be in a relevant place. And then going into the same kind of prediction we made for the UAV data, this is observed grain nitrogen at harvest and predicted. And here's that 20% difference between the UAV and the satellite. So we get pretty good overall linear relationship, but we have to make that additional 20% correction to get our root squared errors and relative root squared errors to similar to what we were getting with the UAV. So there's something going on there, when we're transferring from the satellite, from the UAV at the plots to the satellite. And I think it's probably related to spatial variability. It would be my guess. I'm happy to hear what other people's thoughts are on that. But there was a good study from a group out of Nanjing University who does a lot of drone work. You guys are probably familiar with some of their work that they were presenting at the conference last week as well. They went through and measured nitrogen content at the different levels of the canopy, related them to the drone measurements, and they had a different model. But basically, they came up with a similar spatial distribution relationship with their UAV measurements as I'm seeing when we go from the drone to the satellites. And so I think when we have 40% of our predictors are spatially oriented, and you go from 10 or 20 centimeters to a sentinel pixel, you're changing the surface of prediction pretty dramatically. You're probably getting less vertical variation than you would be getting at the UAV scale. So that's my guess, but we haven't actually had a chance to test that yet. But basically, the scalar differences that we're seeing are pretty similar to what they were measuring when they just did the very top of the canopy compared to the deeper levels of the canopy. So this is some of the other data that you all collected, where on the x-axis, this is the predictions from Sentinel for nitrogen concentration or for biomass. And then on the y-axis, this was a direct comparison at anthesis or a direct comparison at harvest. And for the biomass prediction, I just used a book value for a harvest index to correct to yield. But basically, good linear relationships, but over predictions. And the over predictions are greater at the anthesis measurements than at the harvest measurements. And I would love to talk about why and what you guys think about that. Because it seems like there's promise there, but all the details aren't figured out yet. And just real briefly, but I want to leave a little time for discussion, we are also applying this because we have a lot of genotypic diversity, and creating phenotypes is something that is an important objective for us. So we have been applying these same remote sensing, non-destructive measurements across nitrogen rate trials where we didn't measure the actual things in the season. But we're seeing really good differentiation. This is one season across 4 rates. This is another season across 4 rates. And in a typical nitrogen rate trial, the year effect, the site year effect is a huge impact. But the nitrogen nutrition index is kind of normalizing that on its own already. So that environmental effect is really being reduced in these trials. And importantly, picking out genetic differences. So this is a knockout gene that confers more or less resistant starch across durum background, 2 common wheat backgrounds that are very different in their overall background, but they all are similar in that knockout gene. And we're basically be able to detect the presence or absence of that individual gene across 6 genotypes and 2 years across all these individual nitrogen rates. And so I think what we're getting from this anthesis measurement is probably a proxy for nitrogen uptake efficiency. And obviously we have the ability to calculate seasonal utilization efficiency, but those 2 things are not always the same. And so if you can discriminate uptake efficiency from utilization efficiency, then you probably have a little bit more insight about soil nitrogen and soil nitrogen acquisition versus nitrogen distribution, redistribution, and the properties that come alongside that. So we're excited about those applications. Haven't talked about those 2, but I think we've gone close to our time, and I want to make sure that I leave some time for discussion. One of the things I'd love to talk about is how to take measurements like this, compile them, and get ourselves to a place where we could have a public model that is empirically derived, at least partially, and can be measured everywhere. And that, I think, would really stimulate a lot of possibilities for markets to create insurance products, to create ecosystem services products, something that could actually benefit farmers, so that the regulators can get their regulatory information, but that if the regulators and the markets are working on the same target, and if the public entities like universities like ourselves and other people interested in the public good can create a public model that's improving over time, constantly improving, I think we could replace some of the sort of black box approaches that we see out of the ag tech and private industry, or at least supplement it so that we have some public standard. And I think we have within Sentinel-2, I think we have a lot of the measurements we need to be able to do that, at least in wheat and potentially in maize as well. So that's kind of big picture. I would really like to work across continents on that target and see what we could do over the next 5 years to take existing measurements and be able to create our best bet public model for nitrogen use efficiency and have that help stimulate innovation in private industry and ease the regulatory burden on nitrogen use efficiency. So thank you very much for the invitation and for your attention, and I'll take any questions and conversation.
Very interesting. Questions, comments?
Yeah.
Yeah. Let me say on the mentioned that one possible outcome of this is to unify maybe at a world scale this type of measurements, and this will be based on a common dilution curve. Yeah, we want to estimate the nitrogen nutrition index, and I remember, I think you cited some papers by Miguel, that maybe that curve, and you mentioned in the presentation, it's not so universal, and what do you propose in terms of using a common curve everywhere, or adapting the curve, because this will make a totally difference in the final result of the image?
Yeah, that's a key question. And I think that it's hard to know whether we don't have a universal curve because we don't have sufficient measurements that are accurate, or whether there actually is not a universal curve. From a plant physiological perspective, there should be a universal curve because the carbon to nitrogen ratio in C3 cereals is fixed. There could be different distributions in the soil of ammonia and nitrate that could affect nitrogen use efficiency at the cellular level, but that's very small. Big picture, that concept should hold if you have sufficient data to make it robust. I do a lot of field-based measurements. I know that we make lots of measurement errors because it's hard to measure things in fields. And so when you aggregate lots of field-based empirical measurements, I think that you have a better chance at hitting something that is a true curve rather than an experimental artifact. And so that's my assumption, is that Mikowski and CMPITI are onto something, that the combination of a lot of collective efforts will actually lead us to a more universal curve. One of their studies in wheat looked at nitrogen dilution curve using their Bayesian approach under water stress and under nitrogen stress in the same location, co-location, and they found the same curve.
The same curve. That might have been Mikowski.
It's by CMPITI. Yeah, Mikowski's on that paper as well. But they looked at a data set where, so that's a critical question.
Yeah.
I tend to be a practical person though. Even if we're wrong, even if we decide that it's the universal curve and it's wrong, every measurement, as long as it's being calculated relative to that curve, will be on equal footing, more or less, because it's a curvilinear relationship. And so we may be overstating or understating the NNI by 10% or 20% because we got the wrong curve. And maybe we'll say 1 is the right location when actually 1.1 was the right location. Those are things that matter, and they would need to be played out over time. But the important thing is that we're all using the same one.
Yeah.
And if we could improve it over time, that would be even better. So that, I think it has, going back to the way that we approach the research in California, what I'm proposing is, oh, it's not here. Anyway, that's okay, doesn't matter. What I'm proposing is always going to be cyclical. Like we have to have a feedback loop where we start with the goal. We recognize that we don't have a perfect outcome yet, but that if we have the goal of a universal curve, that we can achieve gradually over time more precision at that curve. I think that we could get there with that more data and better combination of data. So I don't think it's sufficient for us to make 18 different curves per week.
Yeah.
Then we'll never have a universal outcome if we do that.
Yes, thank you.
I definitely think the nitrogen nutrition index is the way to go, completely right. With respect to the concept of the universe of the deletion curve, I totally agree. I mean, this is a universal concept. However, we have to remember that there is really the universal curve and the maximum and the minimum. You remember the paper from math? There is a maximum and a minimum, which is not as narrow and as we would like it. It's too broad. And we start working with the effect of draw in the nitrogen nutrition index in the universal curve because of the Australians. The Australians, they told us, be careful because, and they have quite a lot of information about that in Australia, where they show that the concept works, but we have to be more generous with the margins, with the coefficients. And we check that, and we totally agree. And that's when we publish that as well. So, totally agree with the concept, totally agree with the approach, but we have to be flexible with the margins. Not too flexible, of course, but we just published a paper 2 weeks ago, and it's about the nutrition index. And we end up by saying, okay, if we're going to give like a 0.8, so that means the 20% margin to the values we're getting to decide if the crop, the wheat in that case, is deficient in nitrogen or is well, it has enough nitrogen. Because really, when you're in the curve, of course, sometimes your nitrogen nutrition index is 1.1 and the other one 0.9. They are too close. Really, the values are too close. So if you give me 0.8, then you can really take the decisions. And in that case, probably the curve can be universal. You say this is the curve with the certain error. So how wide is the error? I don't know. But I totally agree with you. If we put data together, we may be able to come out with a curve on the envolvente. The surrounding the error. So
Well, one thing
I totally agree.
One thing that I want to say that I didn't say in the presentation is that I have not found the values in the context of the nitrogen nutrition curve to be as robust earlier than food. When we get in that early vegetative growth stage, I'm not getting very robust or meaningful predictions. The decision support tool that I was showing you guys, that is based on the concept of the reference zone. So when we do that with the farmers, we have them put out an area where there's extra fertilizer. And so we have a lot of data where we have just that simple ratio of fertilized with extra fertilizer in the background. That ratio from both the drone and from the satellite is much more robust at picking up the signal than the NNI. Oftentimes in that early part of the curve, the NNI from our model is not very effective. And I think from measured data, you see that as well. That concentration moves around a lot. So I think part of it may be, I'm not sure that using NNI for a farmer to make their decision on the farm when they actually need to know whether to fertilize is the best use of it. I'm thinking maybe waiting until you have the maximum signal. That's probably when the dilution curve is going to be more stable as well. And with respect to the drought, I think those same interactions are likely to be higher early. And one of the main drought effects that we see across genotypes is not actually a physiological difference, it's a phenological difference. Mostly the breeders have been selecting for rates of translocation empirically.
That's a good point.
And so if you have phenology in there, like that last thing that I was showing you, those resistant starch deletion, it has a different nitrogen uptake signature because it has a different phenology. And they didn't mean to make the phenology different. They were knocking out the resistant starch gene. But in the process of doing that, they disrupted the translocation rate. And those genotypes very consistently are about 4 to 5 days apart. And so they arrive at anthesis at a different point. They do their grain filling at a different point. And so if you have phenology in there as one of your predictors, I think it might help overcome some of the differentiation in drought effects, because I think that some of that is phenological.
So I agree about that. This approach, introducing phenology, could help a lot. Yeah. And it'd be great if we could use nitrogen nutrition index a little bit before, not in teasing, but growth stage 35, 37, which is standard elongation, and we are still on time to apply fertilizer. Because there, the nitrogen nutrition index has big advantage over the NDVI, which is that it's really telling you about the nitrogen needs of the crop. With the NDVI, there is always this debate that is, should we apply more to the highest NDVI areas, which is what we call the king, the landlord approach, or should we apply more to the areas with the lower NDVI, which is what we call the Robin Hood approach? And with the nitrogen nutrition index, we completely overcome this debate, which is a debate which is always going on, and it was in Barcelona, the Congress. With the nitrogen nutrition index, it's clear. I mean, you know what is deficient, where the nitrogen is needed and not. So it would be great if the nitrogen nutrition index could be used as a fertilizer recommendation. Growth stage, I would say yes, standard elongation, because it's true that before is very difficult.
Or you could do the two. Like if you had a good model for nitrogen nutrition index, I'm always a fan for nitrogen. Is easy to apply extra, at least in our setting. You know, if
extra, there is no problem.
Yeah, omissions, that's hard. But a positive control is not difficult to accomplish, and we've been able to convince farmers to put a positive control out, and put it out a single bag of urea at about 30 meters by 90 meters will give you lots of sentinel pixels. So if you can say, put that out, tell me where it is with your phone, and I'll give you an image of that relative to the next door neighbor. I've had some takers on that. Farmers are interested in that. And if you had that relative correction alongside some more absolute understanding of growth stage and NNI at that jointing semi-elongation transition, I think you would gain a lot more confidence. And all that decision support tool, we have the strength of response is just based on that relative value. And the concepts are the same. If you have that point of reference, and then you use an NDVI or an NDRE, something that you know saturates, then you use that quality of the NDRE or the NDRE at saturation to tell you that the NNI curve, basically the critical dilution curve, has been achieved. So I think that there's probably new ways to think about joining those two concepts that haven't been explored. As
Yeah,
well.
that's what is called the nitrogen sufficient index, somehow, no? Which is a comparison to this
Yeah.
reference line.
Mhm.
In the Senate, people from the Senate in Mexico,
Yep.
they were really following the approach. I think it's a very good approach to follow. And as you said, many farmers are willing to have a double pass with the
Mhm.
nitrogen problem.
Yeah, Simon did a lot of that work with the green seeker. Yeah.
Right, that's right. So, and what are you using to measure the nitrogen? Well, nitrogen content, which I guess is nitrogen concentration. Which UAV signal are you using to estimate the nitrogen concentration?
So historically, in some of our other, when we're using sufficiency index, we use NDVI prior to jointing, and then we use NDRE after jointing. And so if we're just using that sufficiency index context, we find that the NDRE is superior once the canopy is closed. But prior to canopy closure, NDVI is superior. In this model, we use all sorts of things. Every single index that we found based on those 5 bands was included. And I think there's 78 features in that model. But about 40% of them, about 28 of them were spatial prediction features. So we calculated the standard deviation and expressed it as a coefficient of variation, or we calculated the standard deviation and expressed it at relative, like if it's the standard deviation of the red or the near infrared or the red edge, we expressed it relative to all the other bands. So it's like it's normalized, the variation from the red normalized to the blue, the variation from the blue normalized to the green, the variation from every index normalized to every other index, plus the coefficient of variance from the individual. So it's all spatially derived, but those were important features.
Because that's one of the questions you say, and it's hard to explain why there's this difference between the UAV and the satellite prediction. Both of them seems to follow it linear, so, but they're different from the 1-to-1 line. No? It's always displacement. I don't know what it could be the reason. I'm not sure.
I'm not sure either. The smoking gun to me seems like spatial variation, but I don't know.
It'd be good. We could check that actually, because in these people experiment we have the satellites, but we have the plane.
Yeah.
We also have some drone, but we definitely have the plane with the hyperspectral, and which 28 times 28 centimeters pixel.
Okay. So then you could upscale from that. Yeah, because I was starting to think about, I have a different data set on the farmers' fields where they put the reference zone out. And we have some drone flights, and then Planet has 3 meter.
Right.
And then the SkySat has about.
We didn't use the SkySat.
But I was thinking
the Planet, yes.
Could you get a layer of, but we don't have as good of variation in those trials because it's just a positive control and the farmer. So you guys have much better variation in this. So that would be an interesting thing to explore is starting to make a gradient on the same exact location of pixels and seeing if that is, and you could add and subtract the spatial predictors and see how the spatial predictors change across the spatial scales.
Because we were using some NDVI comparisons. We use NDVI to compare, and really the data from the satellite and the plane were very close. But we didn't compare the bands, single bands. It's interesting. Okay. Well,
yeah, the spatial predictors. We were working on publishing this. I haven't seen that used before. That was just me knowing that every time I walk into a field and see abiotic stress, it manifests as spatial variation. And so if we have pixels that are segmented in an orderly fashion, we have then the ability to calculate some derivation of spatial variability. And when we threw that in, it did matter a lot. And so it's a mixed bag though. Once you start including that, then scaling it becomes more complicated because how big is your sample? What's a good sample size to capture the physiology that's happening? And you have to be aware, but there's a lot of work that you could follow that's been done on that based on the precision ag within a field. What's real spatial variability versus just artifacts.
And your plan to start with phenology, that will be basically based on the degree days of phenology.
Yep, it's very simple models.
Okay, yeah.
Yeah.
So that's exactly what we're doing now.
Do you guys have a phenological model for this part of Spain or for all of Spain that works, that has been calibrated?
That has been calibrated, not really. I mean, we build our own, and now Lundy is working with Broad. We have like about 100 varieties. But I don't think we have a common phenological model.
Okay.
I don't think so. John, do we have? I don't think so.
No. Well, I've been working with GSAT models.
Okay.
And mostly with maize, but
Well, it's been interesting to see how the California models work because I thought when you were talking about California, I mean, it's like you were talking about the abilities because and rain and variety. So
Yeah,
we have really close.
We have some. We've had some Spanish cultivars in our trials before too. If we're thinking about calibrating across data sets too, we had a Lima grain Star and Lima grain Atomo. They're two white. There's another hard white red making wheats.
And what are the names again?
Atomo, ATOMO.
Atom.
Atomo.
Okay. There is one called Antkera. I don't tell you anything. It's working very well. And the other one was it? Was Atomo?
They call it Star. I'm not sure it's
Wheats.
yeah, they're wheats. They're white wheats. They're from Lima grain, and they were multiple. A lot of the data has those in them, so it would be a genetic check if we had common varieties.
There's a question online. Tatiana, do you want to read it yourself? You can open your mic and say it out loud, or if you prefer, I can read it. I will read it then.
Yes, you can read it.
Here you are. You prefer me to read. What limitation factors do you use in your model? And the second question is how your model is correlated with different field management and current crop situation in the field other than nitrogen.
Okay. The first question, what limitation factors do you use in the model? The model is very simple. It's just phenology plus multispectral image taken at a moment. So there's a wide range of phenological moments where that was measured, starting from the early vegetative all the way through the soft dose stage. And then it's just the reflectance off the surface at that moment. So those are really the two factors that are in there. Either it's just phenology and reflectance in those 5 bands are the core measurements. The derivations from the model are multiple, but they're all derived from the same 5 bands of reflectance. And how is your model correlated with different field management? Okay, so I showed you the 2 treatments where fertilizer was either applied or not applied against different genotype groups. I didn't show data where we had terminal drought imposed. And most of the drought effects were happening after anthesis. That's when we tend to ensure our drought treatments are imposed a little bit more anthesis, but we try not to have it be a point stress because that kind of is disproportionate. And the genotypes arrive at anthesis differently. So our terminal drought, we try to hit after anthesis. So the drought effects showed up pretty much like the regular trial, because we try to not have them be limited by nitrogen. So based on the timing of when we were taking that measurement, there wasn't really any other factor to consider other than water in those trials, and the water stress hadn't really appeared at that moment, so it looked like the regular trial. Does that answer your question?
Thank you a lot. And in this case, also, do you have some difference in image processing for reflectance of bare soil?
Oh, no, we didn't. So we do have a calibration panel where we the day of measurement, we correct for the light conditions, but there was no correction for bare soil. I think that likely that comes out in the phenological predictor. There's multiple reasons you can get the same value of, say, red reflectance, and at the beginning and the end of the season, you might get the same value. And that's one of the reasons I think the phenological predictor is effective, is because it can help the machine learning algorithm differentiate between two values that would otherwise be equivalent, but one was recorded late in the season and one was recorded early in the season. So I think that the soil effects are probably also embedded in that phenological predictor within the machine learning process, but I don't know, that's kind of speculation, but that would be my guess.
Is there any other question?
I have one. If I understood correctly, at some point you said that punitive approach is cheaper than incentive approach. Could you elaborate?
Yeah, from a policy perspective, in California, as an example, they can make a rule and then enforce the rule at very little cost. But if they are trying to reward behavior, it takes revenue. And so they tend to make laws that are punitive because the incentive structure has to come from a new source of revenue that within the government budgets is not really realistic. One successful example in California has been the Healthy Soils program, where they've been trying to trade carbon sequestration value and bank that revenue and then return it to farmers in incentive programs. But measuring carbon is very complicated, costly, and not highly scalable. So those programs have been somewhat successful, but there's always more participation desired than revenue supplied. So that they have a small pool of revenue relative to the number of people who would be wanting to take that revenue. And I think that some of the long-term benefits of nitrogen as a focus instead of carbon is that it's easier to measure, and the carbon savings are permanent, happen on a single year, and so they're durable, they're offset, instead of a soil carbon number that could disappear if you change your tillage practices. And so I think that the nitrogen, to me, seems to be an easier regulatory target, as well as an easier incentive target.
Any other question? Well, now we're going for lunch. Mark to the Gabinete. I think it's time to.
Thank you so much.
Thank you very much.